Hello everyone, how are you all? It's Igcse.science.notesz, and today we're going to explain chapter 4, biological molecules. So basically, let's start with listing the chemical elements that make up the carbohydrates, the fats, and the proteins. So basically, carbohydrates, fats have the same exact thing, which is carbon, hydrogen, and oxygen, while the protein have carbon and hydrogen and oxygen, and it also has nitrate. Now we get to state that large molecules are made from smaller ones. So basically, large molecules are actually made from smaller ones. For example, for carbohydrates, starch and glycogen are made up from glucose. Cellulose is also made from glucose. Proteins are made up of amino acids, and fats and oils are made up of fatty acids and glycerol. And for this point, you get to know that the fats and oils are made up of one glycerol molecule and 3 fatty acids molecules. So basically, the one highlighted in light pink is the glycerol. So glycerol, which is only one, and these are fatty acids, and they are 3. And so these are some extra important points that aren't mentioned in the syllabus, so I'm just gonna say it, I'm not gonna write it. You need to know that the simplest type of carbohydrate is sugars, and they taste sweet and they're soluble in water. And one type of the sugar is glucose, and a glucose molecule is made up of 6 carbon atoms and 12 hydrogens and 6 oxygen atoms. And as you can see over here, that the ratio is 1 to 2 to 1, okay? And they are arranged in a form of a hexagon shape, like, I'm not really sure how to do it, but like it's hexagon: 1, 2, 3, 4, 5, and 6. And the glucose is that way that carbohydrates are transported around the human body, and glucose dissolves in blood plasma, which delivers it to every cell, and cells need glucose for energy. So one use of glucose that you have to know is that cells need it for energy, and as well as that it dissolves in the blood plasma, and this is how the carbohydrates is transported to the body. Glucose link together in chains to form larger molecules. For example, in animal, it forms glycogen, and you're gonna know more about this, like glycogen and insulin and these parts, and like later on. But you got also to know that in plants, glucose molecules are linked together in a slightly different way to make starch, and starch is stored in plant cells. It can be broken down to form glucose again when the plants need it. So this is just a brief, like, you have to know these parts, but it's not mentioned in the syllabus, okay? Now we get to know more about the tests. So basically over here, you need to know that the carbohydrates have 2 tests. One is for only starch, and the other one is for only reducing sugars. Reducing sugars, for example, like other types, which is glucose, for example, okay? So for example, starch, the name of the test is called iodine, and the color before is yellow brownish. And if there is starch present, when you put like droplets of iodine on, for example, a potato which is full of starch, if starch is present, then the iodine color will change into blue, black color. And over here for reducing sugars, it's a test is called Benedict test. Benedict test. And this test is blue. And look for color change. In the presence of simple carbohydrates, which is reducing sugars, the Benedict test turns orange to red bricked color. And over here for proteins, there is another test called Purette test. So this is like Benedict solution. Okay, proteins change from blue. The test changed from blue, and in the presence of protein, it changed to purple. For fats, we use something called ethanol emulsion tests, and it's actually like the ethanol emulsion test is a method to determine the presence of lipids, fats, using wet chemistry. Dissolve food in ethanol, then pour the substance into another container that contains water. If there is fats, it will break into millions of tiny droplets, and its color will change to a white, opaque liquid, milky. If not, it will stay transparent. So before it's transparent, and then after it's milky, cloudy, opaque, white solution. And so basically over here for vitamin C, it has another test. It has a test called DCPIP. You can remember its name by the word C and vitamin C. And that's right here before and here after. So basically before, it's blue-black, sorry. Okay, so blue-black, and after it's colorless. When before, for all of them, you need to know that before is negative result, and after is positive 1. So negative is no, and positive is yes. So here, no vitamin C found, and here, yes, vitamin C found. Okay? Quick revision before we complete, which is a brief summary. Carbohydrates are made of the elements C, H, and O. Sugars such as glucose have relatively small molecules, and starch and glycogen and cellulose are made of many glucose molecules linked in a long chain. Starch can be detected using the iodine test. Benedict solution is used to test for reducing sugars. Fats and oils are also made of the elements C, H, and O. Another name for fats and oils are lipids. So fats and oils have another name, which is lipids. And remember this one because we're going to use it in the enzymes chapter, the one after this one. A lipid molecule is made of 3 fatty acids and 1 glycerol. Also, you need to know how to write glucose, glycerol, and glycogen correctly. Like, don't mess up in the exam because they'll if you wrote glucose in the place of glycerol, they will consider it wrong. Okay, so the ethanol emulsion test is used to detect the presence of fats and oils, and proteins contain the elements C, H, O, and nitrate, which is N. Okay, a protein molecule is made of many amino acid molecules combined in a long chain. So basically proteins is a lot of amino acids. So many amino acids make up protein. Okay? This is a very common question, so please remember this one. whatever. Okay, so basically right now, there are 20 different amino acids, and their specific sequence in the molecule determines the specific protein that is formed. For example, let me explain this part. This is different types of amino acids. So when they link together, okay, they make protein F. But this type of amino acids linking together, these are very random shapes. They make protein W. Okay? Okay. So purity reagent is used to test for proteins, and DCPIP is used for testing for vitamin C. Now we're going to talk about the DNA. So let's now talk about DNA. And basically DNA is the sequence of, it's the genetic material of all living organisms except for viruses, and it has a sequence, and this sequence is that A combines only with T combines only with A, C combines only with G combines only with C. And this is a very common question, especially in paper 2. And this sequence determines that you're a human and not a tree. Okay, and now we got to describe the structure of DNA, and like you need to know that a DNA is made up of 2 strands of something called nucleotides coiled to form double helix. Okay, and that the 2 strands are held together by a bond between the pairs of bases. So it's the 2 strands are held together by bonds between the pairs of bases. So basically over here the shades are not 100% correct. I just made that so it'll be easier. And you just need to know that how to prepare like a test for example starch. For example starch, you need to prepare a suspension of the food sample by shaking a little crushed part of it in a test tube with some warm water. And then you add the 2 drops of the iodine, which is its test, so iodine solution. So when you have a positive result, it's blue-black, and blue-black, which is dark blue, okay? And the negative result should be orange-brown. For the reducing sugar, which is test is Benedict solution, you heat a little amount of the food extraction with an equal volume of Benedict solution in a test tube by placing the test tube in a beaker of boiling water. The positive result is a green, then yellow, then orange, then red, and finally to a brick red precipitate, which is a deposit of carbon 1 oxide, will be formed. So basically, Benedict test, it's not like blue to orange brick, okay? It's blue to green to yellow to orange, then to red, then to brick red. Okay, orange, brick, red. Okay. And now we get to know how to prepare a test for a protein, which is pure test. You add 5 centimeter cubed of dilute sodium hydroxide followed by 5 centimeter cube of 1% copper sulfate solution to a little amount of food extraction. The positive result is a purple color and it should be produced, or the negative result is the color remains blue. So basically you just add the pure test in the little amount of food extraction. If it's a negative result, then no, then it's blue. If yes, which is positive, then it's purple. For ethanol, we said it goes from transparent to milky opaque color. And so you shake 2 drops of the extracted food sample with about 5 centimeter cube ethanol in a dry test tube until the fat dissolves. You pour the solution into a test tube containing a few drops of water. The positive result is a milky white emulsion will form, and the negative result is that the solution remains clear. For DCPIP-C, vitamin C, you draw up 2 centimeter cube fresh lemon juice into a plastic syringe, and then you add this juice drop by 2 drop of a 1 solution of DCPIP. Know over here that the lemon juice we used is that lemon contains vitamin C. Okay, then you add the solution of DCPIP, which is a blue dye, and then in the test tube the DCPIP will become colorless suddenly as the juice is added. We will repeat the experiment but with orange juice in the syringe because it's also vitamin C. If it takes more orange juice than lemon juice to dechlorize the same volume and concentration of DCPIP, then this means that the orange juice must contain less vitamin C. And this is all we have to know in this chapter for the tests. Let's now solve some paper 2 questions. Decided to complete explaining first, then we're gonna solve some paper 2 questions. So let's solve May-June variant 1. You're gonna only find 2 to 3 questions each paper. Here we only have 7 and 8. This is for enzymes, and this question is for chapter 3. We're gonna mostly find them in order. So this question says that this is a protein molecule, and the diagram shows a part of protein molecule, and this is the protein molecule. So these are the smaller molecules that make the protein molecule, and we said that fatty acids, no. Glycerol, no. Sugars, no. It's amino acids. The structure of DNA involves 2 strands coiled together to form a double helix. Which pairing of bases between 2 strands is correct? We said A forms with T, T forms with A, C forms with G, G forms with C. As C and G looks like each other, they form together, and this forms the word at. So at G. Just remember it this way. So A and G no, A and T right, C and T no, C and T right and wrong. So it's only A and T. Now let's go for variant May-June variant 2. We only have this question over here for DNA. This is alimentary canal and maltose and stuff. And over here is the alimentary canal too. We can solve both 7 and 8. So basically, for 8, the one we just said before, it looks like G, so they form together, and A makes the word at. So we saw over here C and G. And this one says that the diagram shows the human alimentary canal. It looks like it's chapter 7 anyway. Which structure is most glucose absorbed up into the blood? It's actually C, but it looks like it's chapter 7. We're gonna have like videos for only solving inshallah, so we're gonna solve these up. This is May-June variant 3, and we're having these 2 questions. Yes, only these 2 questions. So the table it's not A, neither B. And no for iodine. Iodine was for starch. So no starch as well. So it's basically C, but let's check. Benedict's test is for reducing sugars. And Bayer is for protein. Purette. Anyways. So protein and reducing sugars, as we said, both of them are correct. It's letter C. Here, which describes the structure of DNA. Each strand contains chemicals called fatty acids. No. Each strand contains a sequence of amino acids. No. Each strand contains mRNA. No. And two strands coil to form double helix. Yes. Now we're gonna have the class paper to solve, which is February or March 2023. We have for here, which is the test for vitamin C, and I told you guys before. So that's the card you see I see over here. What in DNA, what is the rotation? Again, C, which is together, whatever, and A forms the word correct. Thank you so much for watching. I hope you understand. And so basically, I'm gonna be posting the notes I just wrote while explaining in the stories in my Instagram account. So go follow me for seeing them. They'll be in the highlights.